Hi everyone! My name is Ana Tavares and came from the School of Engineering of the Polytechnic Institute of Porto to present my work entitled Molecularly Imprinting Materials on Cork to Remove Antibiotics from Aquatic Environments. My presentation is divided in 4 parts: Introduction, Goals, Methodology and Results, and Final Considerations. Well, antibiotics are found today in aquatic environment arrive from several sources. One of the sources is the use of the antibiotics. And most antibiotics are used for preventing or treating infection in humans. So, in here, it is possible to see anticipated exposure pathway for antibiotics in environment from the antibiotics used in human medicine as source. In general, consumed antibiotics are discharged into the wastewater treatment plant via excretion. After their effluents discharge antibiotics into aquatic environment, which will contribute to a global dissemination, and as a consequence, resistance to antibiotics increase. And this is a public health concern because it leads to higher medical costs, prolonged hospital stays, and increased mortality. And it is urgently needed to develop methods for removing antibiotics. Currently, there are some methods that are used to remove the antibiotics from water, but those methods have high operating costs and are not selective for antibiotics. So the challenge here is developing a material that is able to recognize and bind antibiotics with high affinity and selectivity. Thus, this work pretends to develop selective material based on molecular imprinting polymer by using a molecular imprinting technique. The molecular imprinting starts with interaction between the template and functional monomers, and then these functional monomers copolymerize with crosslinker during the process of polymerization to form a polymeric structure, and then the template is removed from the polymer, which allows to create artificial reception with specific cavity in the polymer. And these materials are also known as mimetic materials. In this work, MIP was assembled on cork, and first, cork was modified with amine groups, then vinyl monomers, in this case it was used methacrylic acid as a monomer, were covalent bound to cork in prepolymerization step. The polymerization occurs in the presence of antibiotics. And as a proof of concept, in this work, norfloxacin was selected here as the target compound. The production of MIP terminates when norfloxacin is removed from the polymer. In parallel, a control material was also prepared, but without antibiotics, also known as no imprinting polymer NIP. All chemical changes made to the cork were confirmed by the thermal analysis method, in which the loss of mass is plotted against temperature, and as you can see, a thermal reaction of each material, since amination, prepolymerization, and assembly MIP, is different from others. This infrared spectroscopy was also used to evaluate the chemical modification on cork. For amination of the cork, an amine sealant was used. An ER spectrum, that is one your right, showed that the peak located this and this wave number are more evident after the modification of cork with amine sealant. This means that amine group was successfully introduced on cork. After polymerization, peak located at this wave number is more intense and the intensity of peaks associated to amine sealant decrease, which means that MIP was successfully assembled on cork. For removal efficiency study, the cork MIP material was added to 6 flash which containing 3 ml of NOR solution with concentration for 20 micromolar to 120 micromolar. The binding ability of cork MIP to NOR was monitored by HPLC, and this figure shows a chromatogram of HPLC analysis of the NOR that is plotted at 278 nanometer for different concentration, and the NOR was detected with rotation time of 3.4 minutes. After rebinding NOR to cork MIP, supernanos that are in flash were used to determine the concentration of the NOR in solution with the HPLC method in order to estimate adsorbed concentration of antibiotic from cork MIP. These results were also compared not only with standard solution, as you can see, that with no adsorption materials, but also with non-imprinting polymer. Overall, the results indicate that cork MIP had good adsorption performance. To conclude, it's possible to say that molecular-imprinted materials was successfully synthesized on cork and present a good adsorption performance and ability to remove NOR from simulated samples. I want to acknowledge the School of Engineering of the Polytechnic Institute of Porto and Center of Biological Engineering from University of the Minho. The project 3C, Cellulose and Cork in Control of Antibiotic in Aquaculture, and my college from Biomark. Thank you for attention.